Basically, my work deals with a lot of environmental wastes, and this was a last-minute addition to my title, but I had waste earlier in my title instead of bio-waste, but then I realized that I primarily work with bio-waste, and the other remediation processes can range from, say, remediating e-waste to a lot of other inorganic substances that are kind of difficult to biodegrade, and it was a little bit out of scope. So this presentation will be limited to addressing some broader concepts of remediating waste and converting it into useful resources within the context of my research. So to give some perspective to the topic, I would first like all of you to think about a big conundrum that is being faced by industries today. Not only do they have to deal with the rising costs of manufacturing by minimizing non-renewable inputs, but they also have to at the same time maximize their outputs to deal with the growing global population, increasing demand for products, as well as the high standards of living. And because of the growing global consciousness towards sustainability and increasing environmental legislations, they also have to not just remediate their waste, but also ensure that it does not pose any immediate threats to the surrounding environment. So one of the approaches towards solving this problem for the industry is obviously consolidation. Like when you talk about integrating manufacturing processes by reutilizing a portion of the waste that the industry is already generating, then it will be at least an approach in the right direction. So if we were to break down the solution elements to this problem being faced by the industrial and manufacturing sector today, it would be feedstocks, or what do you feed in the industry, how can that be made more renewable or sustainable? What are the conversion technologies that the industries are using right now, and how can they minimize the energy as well as the fuel inputs to these technologies and make it more sustainable, as well as dealing with the issue of waste. Not only do they have to manage it to be more environmentally sustainable, but possibly also look at solutions to reutilize it as a feedstock, which for engineers like us translates into a complicated problem. So essentially right now, as consumers, we derive very heavily from the petrochemical industry. It is not just related to only the transportation sector as in gasoline or other fuels that we use, but it also includes several other consumer products. The essential raw material is crude, but it can be fractionated into several different byproducts, which eventually feed the clothing sector, the paints, the chemical sector, the plastic sector, as well as even food products. So what is the immediate replacement to this highly sustainable platform? So rewinding back a few million years, essentially where did all this crude oil come from? So it is biomass. All crude oil was essentially biomass at some point of time in our history, and due to over the years of extensive pressurization and deposition in like the lower layers of the earth, the high compression changed the carbon of this biomass into the carbon that we see as fuels right now. So yes, basically if we were to project a future where the petrochemical platform is replaced by the biomass platform, then we can hope to have a sustainable and a green future. Although this endeavor is not without its shortcomings, and there are several critiques of there are several critiques for people like us who advocate the use of biomass platform. One of their biggest reasons is that it is very easy to transport crude oil from one place to another because you know it will not change its form. But biomass is not the same. It is also resource limited. All areas of the world do not have the same amount of biomass. And secondly, biomass is prone to degradation. It cannot be transported with as much ease over long distances like crude oil. So these are some of the aspects that kind of limit the use of or kind of limit the authority by which one can say that the petrochemical industry can be replaced immediately by the biomass industry. So why then, what are the positive aspects which are in the biochemical platform that the petrochemical platform cannot receive? The most important thing is, with the help of biological feedstocks, we can achieve circularization of the economy, which is in contrast to the current linear make, use, dispose that is adopted by industry today. With biological sectors, it is possible to make use of the waste that you are generating, extract useful materials from it, and refeed it back to your own process. Also, circularization of the economy helps in integrating seamless different sectors. For example, the agricultural sector, or for example, if we take the food industry that generates waste cooking oil, that waste cooking oil can be reused, say it can be reused as a fuel to feed the transportation sector. Or even if somebody incinerates their waste and generates ash, then ash can be used as an additive in several the construction sector or construction industries. So if we were to ultimately have like the whole industrial sector transforming its platform to the biochemical or the biomass-based platform, then it will be easy to achieve a circularization in the economy, because the bio-based economy is inherently circular in nature due to the nature of the feedstock. And the potential benefits for using is that not only it will reduce our dependencies on non-renewable fossil resources, but also landfills are a big problem in terms of the decreasing land space availability for our world. And also there are several new markets developing for different types of bioproducts. So there's an increasing demand for not just biofuels, but also bio-based chemicals, or everybody prefers organic based chemicals over inorganic substances. So there are several markets that are emerging that need products which can be supplied by this particular platform. And also, if we look at this circle as a whole, then these are what are known as brownfield operations. When I mean brownfield operations, it essentially means setting up an infrastructure that can be readily integrated with an existing industrial platform. So in a sense, if you add a say unit operation to an existing industrial network, then it will also lead to the generation of new job opportunities for the local community. So this, from a social and community point of view, circularization of the economy is also very important. So now I'll get a bit more into bioprocessing. It's not that bioprocessing is a new term. Everybody is quite familiar with it. We have been hearing about bioethanol production, biofuel production for like quite some time now. But all the commercial bioproducts that have made inroads in the market are right now derived from what we call as first generation biosources. These are starch based crops like corn or sugarcane. And bioethanol from sugarcane in countries like Brazil is the prices are as competitive as gasoline, as we'll see in the coming slides. But there are several limitations to using these first generation sources. And once so essentially the starch in these food crops is first converted to sugars, and then those sugars can be converted into several molecules like alcohols, acids or esters, and these are the platforms from which our other consumer products are derived. But the problem for using this is the food versus fuel debate. If we divert food-based crops like corn and sugarcane towards production of fuels, then essentially you cannot feed or sustain your global population, and this cannot be done at the cost of like starvation in developing countries, where you divert more of these crop-based resources into bioproducts. So this is a major factor. And also, if we compare it with the cost of gasoline right now, so there is a tool which is known as life cycle assessment. So essentially, people who model the production of fuels right from the starting point, as in from harvesting corn to like how much you sell the bioethanol for. And if you closely look at the graph, then we can see that the overall cost and also environmental benefits with respect to like greenhouse gas mitigation, land usage or water footprint can only outcompete gasoline if you have a very high biomass source, say ethanol and sugarcane. But if you compare it with the other cases, then this process is not very competitive as compared to use of conventional resources like gasoline. So the life cycle assessment results are quite negative, because even if we think about bioethanol production from biomass, then the amount of fuel that goes into, say, transporting your crop from the field to the factory where you ferment it to produce ethanol, that also involves fuel input, which is taken into account for the greenhouse gas mitigation. So then we need to shift to what are known as second generation resources. Second generation refers to lignocellulosic biomass. Lignocellulosic can be broken into lignin plus cellulose. This is essentially the part of the biomass that we don't utilize. Say if you talk about corn, then the corn stover will be the second generation resource or the lignocellulosic biomass source. So this can range from agricultural residues, forest residues, or what are known as bioenergy crops, crops that are cultivated just for their foliage or the lignocellulosic content and can be converted into biofuels, and they don't create any food security issues. And also cellulosic waste that includes bio waste, municipal organic waste, as well as paper waste and food processing wastes. So this is a particular crop, sorghum, that I worked with in my undergrad. It is being increasingly recognized as a replacement to sugarcane because sweet sorghum stalks are as juicy as sugarcane, and it is kind of easy to convert it into bioethanol. So this cultivation has been taken up by several prominent companies and research institutions. Although bioenergy crops still have to be cultivated on land. So you are maybe mitigating the issue of food security, but the problem of mitigating land footprint still remains. So then what next? So the correct procedure is then to focus on something that is waste, that is creating a problem for managing it, as well as can be a potential source for feeding the fuel or the bioproducts industry. So now finally I'll get into the somewhat introducing to my thesis topic, that I am kind of looking at such type of waste sources which can be readily converted into bioproducts that the world needs right now. And also if we focus on this graph, then this is a sort of projection for biofuel or bioethanol production from the different type of feedstocks that we have right now. And second generation, it will take time, but the first generation sources will eventually plateau out in the 2020s. This is a prediction by the Department of Energy of the Federal United States government. And second generation biomass, or the lignocellulosic biomass I introduced in the last slide, will become competitive around, say, in the next decade or so. So essentially the research and development section should be ready for it. So if this is such a readily available source and lignocellulose is the most abundant renewable resource on biomass, then why hasn't this paradigm shift happened already? Like, why aren't people already switching over from first generation to second generation resources? This is because lignocellulose is very difficult to break down. If you imagine the structure of reinforced concrete, then that is what the plant cell material is like. Lignin exists there because it was made for a reason, so that plants could have their self-defense against natural attacks or, say, pathogenic attacks. So the cellulose, which is our source of sugar, is essentially entrapped in a lignin matrix, which is very difficult to break down. So first you have to get rid of the lignin, break down the hemicellulose, access the cellulose, and only then you can hydrolyze it to, say, sugars or other molecules of your interest. Whereas for starch-based resources like corn kernels or sugarcane, you already have the starch available, so there is no pre-treatment step to it. So if we go down from feedstock type from sugar, starch, cellulose, although the feedstock cost decreases, but the technical complexity of utilizing these resources increases. So to this end, if we think a bit about an industry that can, say, provide us with a waste lignocellulosic resources that does not require as intensive pre-treatment as the other lignocellulosic feedstocks, then an answer that comes to mind is the pulp and paper industry. In Canada, it is a very important industry For the whole country as well as for the BC province, the pulp and paper products form a big part of its exports, as well as in comparison to the rest of the world, Canada is a very big producer of paper and pulp related products. So the byproduct of this industry, pulp and paper mill sludge, which is the feedstock that I am investigating, is essentially the leftovers from the wastewater treatment of this industry. So it is a waste that is available and represents a big land usage for the industry, because right now they get rid of it by disposing it in landfills. And even though it is essentially waste paper and the other byproducts of the industry, so if they want to burn it to recover energy from the carbon content, and some industries do that, and also to reduce the ash that they send out to the landfill, but the sludge in its original form is very wet. So dewatering and drying it before you can actually burn it to use it as a fuel takes a very high energy input. And so the whole process ends up being a net cost to the industry rather than they recover energy from it. So there is a prospect here to say add value to it, and if it can be converted into a product that can be reutilized by the industry itself, so we would be able to obtain a circular model around this industry as we had seen earlier. So why do I call this an all-encompassing solution is because it is cheap, it is readily available, and if we compare it to other waste lignocellulosic feedstocks, the lignin, which is the thing that we hate the most because it's so difficult to break down, is present in relatively very small percentages as compared to the other feedstocks. This is because when paper is made and before the sludge is generated, due to mechanical pulping, a lot of the lignin already gets deconstructed in that process. So this leads to the sludge having a very low lignin content, and it is very high on cellulose and hemicellulose because it is essentially paper. So during the paper making process, when they pulp the raw wood chips or wood pellets, they want to extract as much cellulose, as much high quality cellulose fibers from it. So they have a length requirement. There is essentially a screen that separates the kind of fibers that you would need for paper making and reject the fibers that don't pass that criteria. So this waste that comes out is still very rich in cellulose. It is just because the fiber length is small, that's why it can't be used for paper making. But for our purpose, for converting it to any other bioproduct, it is a very good resource. And like any other lignocellulosic biomass, there are boundless biochemical conversion opportunities from it. So this is a semi-postulation of a circular economy that can be developed around the paper mill industry by utilizing this waste sludge. Now there is not just bioprocessing that can convert waste sludge into fuel or energy, but there are several other thermochemical routes as well, or anaerobic digestion wherein you produce biogas or methane. That is another way to recover energy from this waste sludge. But within the context of my research, I am focusing more on the bioprocessing aspect, where you use biocatalysts or enzymes to convert this feedstock into the product that you need. So essentially, if we think about conversion of any bio-based feedstock into fuels, the processes that follow are, first of all, you have to pre-treat the feedstock so as to release the cellulose and sugars, as we just saw now. But for paper mill sludge, this other feedstocks usually involve intensive temperature-based or high acid basic pre-treatment requirements so that the lignin breaks down and you release the sugars. But for paper sludge, this should not be an issue. Then downstream to it, the sugars that are released are coupled with some organisms that are capable of converting those sugars into alcohols or any other product that you want. And trust me, genetic engineering can essentially help you manipulate any organism to produce almost anything that you want. So how I will be approaching this issue of converting paper mill sludge into a useful product is by using an approach which is called metagenomics. Now we have bacteria all around us, but all of the bacteria cannot be exactly captured and cultured in a lab so that we can work with them. And the amount of bacteria that we can actually culture is just a minuscule percentage of how many bacterial species are out there. So metagenomics is essentially a technique that helps you capture the other genetic potential which is not culturable in a lab. So if you pay attention to the microbial community pictures, this essentially means the environment from which you can extract any, say, enzyme that you want. For example, I am working with sludge and I'm looking for certain enzymes that will be able to break down my sludge into my cellulose in the sludge to give me sugars. So what approach I am using is that I will extract DNA from the sludge itself and convert it into a metagenomic library and then screen it further for the type of activity that I want or cellulose breaking. So how we do it in the lab is we extract DNA from the environment of interest. We use plasmids, or they are a different type of DNA present in bacteria that helps you combine external DNA with it and then insert it in the bacteria, so that you know that this DNA will now be expressed in the host of your interest. And then you screen the different clones or the bacterial cells that you make in this way for the activity that you want. So some people may just want to see that if this environment has the particular activity that I want. So what they do is a sequence-based metagenomics, wherein you clone the bacteria, you essentially sequence the DNA, and we have databases of sequences that correspond to specific enzymes that we want. So what they do is an in silico or on computer comparison of both the sequences and then analyze to see if this environment has the activity that I want. But if you want to discover some, say, novel enzymes from this environment, then this approach will not work, because this will tell you what is already present and documented. So to get some novel catalytic activity from this environment, the other part of metagenomics is function-based metagenomics, wherein you essentially use libraries of these organisms that are present in a suitable format, and then you screen them for that particular activity. So suppose if I take my organism and put it in an environment where it will generate a color that will tell me that it is breaking down the compound I want. So that is how I will get to know that this organism is capable of the activity. I may not analyze the sequence right away, but I know that this is doing the function that I want, hence the name function-based metagenomics, and then analyze it to see if it's, say, a new cellulolytic activity that I have discovered. Simultaneously, what also other people do is add some compounds that can, selectively let the organisms that have your activity grow in the medium and then kill out the other organisms. So there are several approaches by which you can discover novel activities from these metagenomes. And this picture essentially tells you the process, like you extract DNA, you fragment it into like specific size sequences, and then you clone it into your host and then use this host to screen in whatever metagenomics approach you want to. So now we saw how to get the biocatalyst that we want for the process, but how do we actually define the process? So conversion of biomass to fuel is essentially hydrolysis followed by fermentation. These three classifications are dependent on how hydrolysis and fermentation are coupled with each other. If both of them take place separately, then you get the first thing, SHF, which refers to separate hydrolysis and fermentation, where you have the hydrolysis and fermentation happening in separate vessels. SSF means simultaneous saccharification and fermentation. So here you have a one-pot chemistry of fermentation and hydrolysis happening in the same, essentially, vessel or bioreactor. So there are pros and cons to both of it. If you separate both the processes, then you're able to control them better. But after some time, it is obviously a costly process and it's difficult to scale up because you're dealing with two separate units. But if you combine them, then the product control is less, because if your hydrolysis is happening in a completely different environment versus your fermentation, then it is very difficult to couple the both and find, say, optimum process conditions. And ultimately what bioprocess research is vying towards is CBP, which is known as consolidated bioprocessing, which is essentially a one-pot chemistry of pre-treatment, hydrolysis and fermentation. So this is the ultimate aim, it is very difficult to achieve, but would represent the most ideal case where you have all the processes happening essentially in one pot to give you your final product. So how can we optimize this one pot chemistry if we were to do so? So as a bioprocessing engineer, we play around with certain process variables like the growth medium composition, or say like what type of nutrients does my strain or my bacterial culture need to be able to perform all the functions that I want. What is the specific pH, or say how acidic or how basic is my media that would lead to optimum growth of my culture, whether it is aerobic or anaerobic, how much oxygen does it need to survive, what is the correct temperature, because in chemistry, if we look at how enzymes function, then we'll come to know that enzymes have something called an optimum temperature, below which and above which they are not very active and hence will not catalyze the process I want. So temperature is also a very important variable. And RPM is essentially the how well you rotate your system. So if your system is rotating well, and so you're essentially limiting your mass transfer resistances. If your bacteria are, say, growing as clumps and they are not in continuous contact with the nutrients that you're supplying them, then that will also limit their efficiency. So you have to ensure that the medium is stirred well, or like there is least mass transfer resistance. So when I told you it's not as easy as it looks, this is what an ideal consolidated bioprocessing organism would look like. It will not just have to secrete those enzymes, it will have to hydrolyze the biomass, metabolize the sugars, and also produce the desired byproducts, which would mean engineering a lot of different metabolic pathways in one single organism. And also, if the organism does all things, but at the end, the product that it produces is inhibitory to its own growth, then it cannot tolerate high concentrations of the product that you want. And if you were to scale up this process, then that would be very detrimental, because we want to maximize the yield of product from our process. So this is super difficult. But there are certain approaches in genetic engineering that can make you get around with this. And there are pros and cons to both. If you were to do this in a single organism, as we observed earlier, then it is very difficult. But again, because you are dealing with only one strain, then it is kind of easy to manipulate it. Whereas there is a second approach which is known as consortial engineering. By consortia, we mean essentially a group of microorganisms. If you were to divide this very difficult metabolic loads into two different strains and then make them coexist together, then it would be, after all, a bacterial cell is a living and a dynamic system. If you engineer too many, say, control points into a single machine, it may break down eventually. But if you were to divide that, the control points or the processes that you want into two different strains, then they may function like better, as in the strains would grow better. But again, the challenge here is optimum control. If you were to just like divide, if you know that one of your strains does good hydrolysis and the other strain is a good fermentative organism, but there's no guarantee that the two will coexist very well. So you don't have to only like genetically engineer the two strains independently, but you also have to engineer the consortia as a whole so that you have control over what you want from the consortia. So in relation to what we just discussed now, like overcoming or getting a better output from if we were to use, say, a consortia, then now I'll talk about a bit of the challenges that we can face by using this feedstock. Anything is not without its own drawbacks. So the pulping and paper making process, although deconstructs lignin, but adds a lot of sludge and other toxic chemicals to the sludge, which is very detrimental for growth of microorganisms, because all these heavy metals, as you can see as silicon, aluminium, iron, they get converted to ash part. They are essentially represented as ash and can be a toxin to the microorganisms that you want to use to convert this sludge into, say, any bioproduct that you want. And even if I'm extracting the metagenomic DNA from this particular sludge and cloning it into another microorganism that is easier for me to work with, I randomly fragment the DNA. So there is no guarantee that the new microbe that I have will be able to survive in this particular environment as compared to a microbe that is like isolated as such from the environment. So we have to think about getting around this particular drawback also. So aiding in this approach this is a sort of transcriptomic based algorithm that I've formulated for my screening steps. This screening is different from the catalytic screening. This will now be a screening for survival in my medium. And I will not, I don't only have to see that if it is able to, if my strain I know is cellulolytic but does not works well on pure substances like say pure cellulose but is not able to survive in my own sludge medium, then it's of no use to me basically. So in order to genetically engineer what are known as sturdier clones that can be cellulolytic as well as survive in this medium, we use techniques that are known as transcriptomic techniques, and specifically I will be using what is known as subtractive hybridization. So to boil it down, it is just a mathematical addition and subtraction technique. You isolate your mRNA from your toxic environment, which is my sludge, and you again isolate the mRNA from a metagenomic strain that you have developed from that sludge. You mix both of them together. The initial mRNA is labeled with a certain marker that distinguishes it from the mRNA that you isolate from your metagenomic library. We hybridize them. By hybridization, I mean that the common areas of both the mRNAs will fuse with each other. And then you use certain techniques to separate out the fused or the hybridized mRNA with the mRNA that is not hybridized, and you isolate that mRNA. So this difference of the two fused mRNAs is postulated to contain the survival genes that we would need, and then I would convert this mRNA into DNA, reclone it back into my own clones, and then test to see if this gives it the survivability capability that I need for my own process. So you basically add them together, take the common parts out, and you subtract the differential mRNA and use that to create your own sturdy clones, which will not just produce sugars but also be able to survive in your own feedstock and essentially use the toxic waste as a feedstock. So my research problem definition is essentially, is it possible to devise a consolidated brownfield process for adding value to this particular feedstock by utilizing it not just as the resource for converting to bioproducts but also as the source of the catalysts that will bring about this process change. And my hypothesis behind choosing this is because it is a cellulose rich medium. So I'm hypothesizing that the microorganisms that are capable to survive in this medium already have the activities that I want or I'm looking for. And this essentially would lead to circularization of the economy around this very important industry in Canada as well as BC. And it can be converted from a source of net cost for the industry to a potential revenue source, depending on what downstream process we convert it to. So the novelty in my research is essentially that cellulolytic activity has been mined from different other environmental sources. For example, the rumen gut, because we know that ruminatic animals like cows eat a lot of cellulose, so their gut is postulated to have those type of microorganisms that are capable of breaking down cellulose. Also forest soils or even peat coal beds, these are sources of microorganisms, but a metagenomic approach to paper mill sludge has so far, I could only find one work, and also it was from a different part and not exactly what I am targeting. But you cannot shoot in the dark. I need to have some literature to back my hypotheses as well. So people have prospected paper mill sludge for the type of activities that I am looking for, but their approaches have been different in a way that they isolate single organisms from this sludge and then basically postulate that these have the particular cellulolytic activities. What you can see here are bacterial growth plates. So you can spot a region of clearing on those plates. That essentially means that your bacteria that is growing is able to eat away the medium that comprises of cellulose, and hence we say that these bacteria are capable of breaking down cellulose. But the problem with isolation and enrichment based approaches is that they are very low throughput. So you cannot screen your own environmental DNA at the time, and it's also like kind of a luck basis. If you're able to you will isolate a lot of microorganisms from any organic source, but to what extent or how many microorganisms will do what you want to is essentially what makes these processes low throughput. And the metagenomics approach that I am taking is essentially a more random approach, but because your sample size is very big, so your probability of success increases in a way as compared to the isolation approaches. And my targeted end product is butanol, which is a different biofuel as compared to bioethanol. It is better in many ways. It is less corrosive, less hygroscopic, does not absorb as much moisture, is very good for blending. And bioethanol cannot be used in the engine, the type of engines that the transportation industry uses right now. But because butanol is a bigger alcohol than bioethanol, is more similar to gasoline, so this has the capability of being used directly in the engine without any need for blending essentially. So in this way, it is much better than bioethanol as a fuel. But the challenge for producing biobutanol as well as using it as a fuel is that while bioethanol has a straight metabolic pathway where you can easily use any fermentative organism, say baker's yeast or E. coli, to produce bioethanol, biobutanol has a slightly trickier mechanism to produce. The organism that naturally produces biobutanol produces several other byproducts, from which biobutanol is, say, one of the major products, but optimizing its yield or forcing that organism to produce only biobutanol would compromise on its growth ability. So, metabolically engineering this pathway into a more user-friendly host like E. coli is still challenging and in the research and development phase, and the yields that are produced, that are in literature right now, are not very competitive with the industry, unlike bioethanol, which has already been commercialized. So, to end, I would like to just throw back the perspective to you that what is bio-waste for most of us, and unlike some people who are environmentally challenged or whatever, but bio-waste for an environmental biotechnologist or engineer like me is a revenue source, is a source of potential resources that can be converted into products that the consumer market needs or requires, ranging from fuels to chemicals to even biopharmaceuticals, and kind of like my bread and butter. Yeah. So, but the very first thing, before even we think about changing the society's perspective of viewing waste as a resource and not as garbage, we have to essentially rethink the technology behind how we convert it. When people talk about bioremediation, they simply view it as a means to make waste less hazardous or make waste in a way that can be easily disposed in the environment. Still, there is a big school of thought in bioprocessing that does not view bioremediation as a bioprocess in itself or something that can add value to the waste. So this paradigm shift has to first start from the technology onwards. When you devise technologies that can show the society that your waste can be generated into something of use, then only it will foster confidence in them. So this paradigm shift has to start from the technology itself. So earlier, remediation was limited to, say, cleaning up oil spills or incinerating your waste and disposing it in a less hazardous way, as compared to the current school of thoughts that are developing to be integrated resource recovery, where you combine remediating the waste to adding value to the waste. The first picture in the now box that you can see is biochar. Biochar is essentially something that is derived from bio-waste by using different thermochemical routes, but it is a value-added product because it is used to better your soils for plant growth or also as a fertilizer. Initially, sludge used to be just remediated and disposed, but now it is widely recognized as a source of biogas or essentially fuels. And so as long as technology is able to convert organics into something that can be a potential source of revenue for society, it will be easy to bring about the shift that we want. So a couple of like close home examples that can like derive home this fact is Metro Vancouver, like our very own municipality, is taking a lot of endeavors to recover nutrients from the waste. So the wastewater treatment plant that are in and around Vancouver have started integrated resource recovery to not just treat and dispose water or sludge, but also recover fertilizers or nutrients from it, as well as use it for energy generation to meet part of the energy requirements that the wastewater treatment plant requires. As well as there are other examples in the US, or a very recent example that I came across in one of my classes is this group called Janaki Bioenergy, which has essentially set up a technology for converting human waste into or obtaining drinking water from human waste. And that is Bill Gates, who's actually trying the sample himself. He was the one who funded the whole research, and obviously any endeavor like this needs investment from the other, say, entrepreneurs or society in general. But if people truly believe that waste is essentially a treasure, then it will be easier to develop such new technologies, because technology, apart from science, also needs resources to develop. So we have to be conscious of that fact. So my research implication is essentially pertaining to the broader concept of developing a zero discharge paper mill model and addressing several interdisciplinary challenges across environmental engineering, metabolic engineering, developing a nexus between energy and waste management, as well as fostering a circular bioeconomy development. So, okay, if you didn't understand my presentation so far, I have a video for you.
Perception is a powerful human emotion. How we see things can change our lives and the world. We see this bin full of books as a treasure, while this bin full of waste paper as garbage. The circular economy will help us realize that this waste can be shaped into new books, clothing, or even biofuels. In the future of the circular economy, waste shall cease to exist. Industries as diverse as food and transportation will fuel each other's growth through systematic integration. Power will not be bought but generated from any and every byproduct. Communities will grow and prosper in a holistic manner. My graduate project is about establishing proof of concept of a zero discharge paper mill. One of the biggest drivers of economy of British Columbia, this industry currently incurs an annual cost of around $5 billion landfilling its waste sludge, suffering economically and environmentally. Through bioengineering, I will use this waste as a feedstock in a consolidated process to generate a chemical that can be fed back to paper making itself. Commercial scale-up of this circular model will reduce process economics and generate employment. It will hence serve as a powerful example, fostering similar endeavors towards a circular and sustainable future.
Okay, thank you.